All right, so remember, we are looking at mutation. So let me recap. Mutation is basically a result of an alteration has taken place on either a gene or alteration has taken place on a chromosome. All right, although gene and chromosomes are related, they are not the same. A gene is a small unit on a chromosome. A chromosome is the entire chromosome. All right, so here, this whole thing, follow my finger, this whole thing, this whole structure is a chromosome. But in the chromosome, that small shaded region which I've done, and that's a gene. All right. So like that, you have many genes. On this particular chromosome, you have many genes. How many genes do you have in a chromosome? All right. You can have as many as 500 genes on a chromosome to 1,000 genes. All right. So in one particular chromosome, you have 500 to 1,000 genes. All right. Okay. So what is this mutation all about? First of all, let me tell you, there are two types of mutation. One, gene mutation. So gene mutation obviously only involves the gene, not the entire chromosome. That means gene mutation only involves this particular shaded region. Therefore, something in this shaded region has altered, has changed, making the gene not to function normally. The gene is no longer normal. The gene is abnormal. Now, sometimes the abnormal, being abnormal, it can give you a good characteristics. Then it is a favorable mutation. In humans, no. In plants, yes. In plants, we have come across a lots of favorable mutation. But in humans, no. In humans, mutation is always seen as something that's bad. It always causes disastrous effect. So far, we have, so far as mutation in humans is concerned, it is considered as bad. Now, sometimes the mutation affects the entire chromosome. It affects the entire chromosome. It affects the entire chromosome. That is called chromosomal mutation. So we have two types of mutation, gene mutation and chromosomal mutation. So now I'm going to focus on gene mutation. So I'm going to focus on the shaded region. So this gene shaded region, I am going to blow it up. So when I blow it up, you will find that it is made up of nucleotides. So I'm going to put that nucleotide 1, nucleotide 2. I don't want to draw the nucleotides, okay? Nucleotide 3, and so on and so forth. So you have many nucleotides, okay? Now each nucleotides are linked together. Each nucleotides are linked together. So they all are linked together. Okay, by a phosphodiester bonds and all that, you don't need to worry about that. Now, in each nucleotide, there is a nitrogenous base. There's a nitrogenous base, there's a nitrogenous base, there's a nitrogenous base. Now, the sequence of the nitrogenous base. So let's say this is nitrogenous base 1. For example, the nitrogenous bases are adenine, guanine, cytosine, all right, and thymine. So this can be adenine, this can be adenine, this can be guanine, it can be cytosine. All right. So the sequence of the nitrogenous bases. So let me simply put it as N1, N2, N3, N4. This sequence determines the function of this gene. It determines the function of the gene. All gene has a certain function. In fact, they determine, all right, one of the functions will be to determine the type of protein that is to be synthesized by the ribosomes. That's one. Let's not go into that. You just simplify your idea. Just think of it as a function. All right. So what determines the function of this gene? The sequence of the nitrogenous bases. So after 1 should be 2, after 2 should be 3, after 3 should be 4. Now what if I take this 4, this nitrogenous base, put it here, and the 3 comes there? So which means I've altered the sequence. It is the same nitrogenous base, but I just altered the sequence. Instead of 1, 2, 3, 4, it is 1, 2, 4, 3. So when that happens, your gene do not function normally because it is the sequence of the nitrogenous bases. That means not only what type of nitrogenous bases, how many nitrogenous bases are in the gene. Some genes you have 20 nitrogenous bases. Some genes are made up of, usually it's hundreds of nitrogenous bases and so on. All right. So you cannot just say, all right, the number of nitrogenous bases. You can say number, you can say type, you can even say sequence, all this. So let's stick to sequence. So the sequence of nitrogenous bases must be maintained in order to cause the gene to function. So coming back, when you say gene mutation, what do you mean? One, the gene mutation got 4 types. There are 4 types of gene mutation. All right. One type of gene mutation. Let's stick to this 4 and let's explain. Let's stick to this 4 and explain. So one type of gene mutation will be where the nitrogenous base 3 is completely deleted. Then somehow or other something occurred. So due to harmful, usually mutations are caused by harmful radiation such as X-ray, gamma ray, exposure to carcinogenic substances. So let's say this person is exposed to one of this, either the harmful radiation or carcinogenic substances, and as a result, like this nitrogenous base, this whole nucleotide, we got the nitrogenous base. This nitrogenous base has been deleted. So the sequence from hold on. The sequence from 1, 2, 3, 4. All right. The sequence from 1, 2, 3, 4 is now 1, 2, 4. Two things have happened. Number one, the nitrogenous base has been deleted. Nucleotide, the nitrogenous base has been deleted. Number two, it has altered the sequence. This is called deletion. So this is called gene mutation, and this gene mutation is referred to as deletion. All right. So deletion is one type of genetic mutation. So what has happened to the deletion? One of the nitrogenous bases has already been deleted. One of the nitrogenous bases of the nucleotide has already been deleted. It's gone. Now, is that all? No. In certain cases, the exposure to these mutagens, which are the harmful radiations of that, can cause a substitution of the nitrogenous base. That means this nitrogenous base, together with the nucleotide, is removed, and a new nucleotide has come in, or just the nitrogenous base has already been removed. And because why? The component of a nucleotide is what you call the component of nucleotide is 3 phosphate group, pentose sugar, and nitrogenous base. The pentose sugar and the phosphate group is the same for all nucleotides. That is why we never speak of that, because I say it is only the nitrogenous base that is different. So let's stick to the nitrogenous base. So this one, N3, has been removed, but it is replaced with N5. So the sequence is 1, 2, 5, 4. That is also wrong. That also makes the gene become abnormal, non-functional. All right. But this is called substitution. This is called substitution. So the second type of gene mutation is substitution. First was deletion. All right. Is that all? No. There's another one. All right. What is the other one? Okay. So instead of what I told you, instead of this being substituted by N5, the reverse. That means N3, N4, they exchange positions. They exchange positions. So now it becomes, that is the same thing there. Now it becomes 1, 2, 4, 3. 1, 2, 4, 3 instead of 1, 2, 3, 4. So it's just inverted. 3 and 4 has inverted their positions and exchanged their position. Now, will that be okay? Answer is no. The sequence is extremely important. Even a small change between 3 and 4 will cause the gene to be non-functional and can give you disastrous effect. This is what we call inversion. So this gene mutation is called inversion. So I've already explained to you about substitution. I've already explained to you about deletion and inversion. Finally, now you have 1, 2, 3, 4, and a new new nucleotide with its nitrogenous base comes and fits into here. So instead of having 1, 2, 3, 4, you have 1, 2, 3, 4, 5. All right, you have an extra nucleotide. Does that make you a superhuman being? No. See, the thing is such, you can't have extra, you can't have less. you must have exactly the same nitrogenous bases in the same sequence. That's the point you should grasp. You should catch that strongly. So even if a new nitrogenous base is being added, that will still give you a non-functional gene. And that is still gene mutation, but it is called addition. Therefore, gene mutation, there are 4 types. All right, what are the 4 types? You have deletion, you have substitution, you have inversion, and you have addition. All right, this is all happening where? It is all happening to that particular unit, inside that particular unit, involving only the nitrogenous bases of the nucleotide. It is not subjected to the entire chromosome. That means that part of the chromosome, the genes on that part, the genes on that part, none of them are touched. None of them are disrupted. None of them are disturbed. Therefore, they can carry out a normal function. But only that particular gene is being affected, involving the nucleotides inside that. That is what we call gene mutation. All right, so I hope you understand very clearly. And now we can go to our notes and fill it up. All right, okay. You can take down whatever you want to take down. Huh? All right.